The ABO and RH systems are defined by genetic architectures on chromosomes 9 and 1, their complexities and gene deletions transforming clinical medicine. In 1937, a discovery was made. Inside the blood of the rhesus macaque, scientists found a brand new antigen, an inherited protein on red blood cells. This is the rhesus factor.
Landsteiner discovered blood groups A, B, and O. DiCostello and Sturley found group AB. Proof of inheritance followed shortly after. Bernstein proposed the three-allele model.
The modern synthesis unites genetics with evolution. Mapping reveals the precise architecture of DNA, charting the code of life.
Mapped to chromosome 9q34.2, the ABO gene features 7 coding exons. It encodes a glycosyl transferase, modifying the H antigen.
The A allele produces A transferase, transferring GalNAc to the H antigen. The B allele transfers D-galactose, while the O allele is inactive.
Located on chromosome 1p36.1, RHD and RHCE are paralogous genes. Each contains 10 exons in an antidromic orientation, sharing roughly 98% homology. The RHD gene encodes the D antigen. Its total deletion is facilitated by flanking rhesus boxes. These boxes undergo homologous recombination.
The A and B alleles diverge at 7 positions, 4 alter amino acids, notably XT6 and 268. In BAC, in B, methionine and alanine favor UDP-Gal.
Variation is driven by A1 and A2 alleles. The A2 allele has a substitution at 467 and a deletion at 1061, creating an extended protein with a reduced catalytic rate. The RHCE gene is highly polymorphic. For C/C, position 103 is critical. EE relies on a single nucleotide change. The ABO blood group system is mapped to chromosomal location 9q34 tier. The ABO gene encodes a specific glycosyl transferase. This enzyme uses the H antigen as its primary substrate. The RH blood group system is clinically vital. Located at chromosome 1p36.11, it involves the RHD and RHCE genes. These produce structural transmembrane proteins.
Under the HH system, the FUT1 gene is located. It resides at chromosome 19q13.33. This gene produces fucosyl transferase. Genotype is your genetic makeup, while phenotype is the expressed trait. In the ABO blood system, A and B alleles are completely dominant to O. However, A and B are codominant. RH inheritance is modeled as a single gene trait. Rh positive is dominant, while Rh negative is recessive. The D notation signifies the absence of the RHD gene. Predict offspring blood types using a Punnett square. Cross an AO parent with a BO parent, and OO genotypes. That's all 4 major blood types from just these parents, perfect for clinical risk checks.
An AB and O cross produces a genetic paradox. No child will share either parent's blood type. When both parents carry the recessive Rh trait, there's a clinical concern, a 25% risk.
Antigens are key components of the cell membrane. Often made of glycoprenes or glypids, they project from the surface specific molecular ID tags. This allows immune cells to recognize them.
Rh proteins form a complex providing stability. It is tethered directly to the cell cytoskeleton. Without it, red cells become bowl-shaped and fragile. These cells suffer a shortened lifespan. Beyond support, blood group proteins have functional roles. Rh proteins are similar to ammonium transporters. The complex facilitates transport of ammonia. This connects genetics to systemic metabolism. Blood group genes show pleiotropy. Non-O groups have higher clotting factors, increasing thrombosis and infarction risk. Type A is linked to specific cancers, where losing antigens is. Malaria has shaped the human genome. Type A and B cells form dangerous rosettes. Type O cells resist this, saving lives. Pathogens have shaped our blood type evolution. Cholera made type O blood a severe liability. Norovirus uses these antigens as receptors. H. pylori strains adapted to host demographics. For over 20 million years, primates have shared the A and B blood alleles. This indicates a process called balancing selection, survival advantages against fluctuating diseases.
Blood groups reveal ancient human migration. In South America, type O approaches 1, type B is most prevalent across Asia, while R negative peaks in Europe, tracing our shared global ancestry.
Human blood is classified into 4 main groups. These are A, B, AB, and O. We also see distinct distribution patterns, tested across both male and female genders.